Hello students, welcome to EPG Pathshala. I am Dr. Riddhi Garg from Deshbandhu College, University of Delhi. Today we are going to talk on module Microarray Part 2 from the paper Molecular Genetics. The following are the learning outcomes of the module. You will be able to learn about different types of microarray, know how different types of microarray experiments are performed, evaluate the importance of microarray in research, know the current and potential applications of different types of microarrays. Microarray is a new and robust technology that has driven research in the field of molecular biology to great heights. In Module 30, Part 1, the concept of microarray was introduced and DNA microarray was discussed in detail. But there are several different types of microarrays that exist and are playing a very important vital role in research in several fields like immunology, proteomics, enzymology, etc. In this module, we will introduce the basic concept, working, and uses of the different types of microarrays such as protein microarrays, peptide microarrays, tissue microarrays, cellular microarrays, etc. Types of microarrays. There are several different types of microarrays like DNA microarrays, protein microarrays, peptide microarrays, tissue microarrays, cellular microarrays, chemical compound microarrays, antibody microarrays, carbohydrate microarrays, and phenotype microarrays. Now, in Module 1, we have already discussed about DNA microarrays. So, in this module, we will be talking about the rest of the different types of microarrays. Protein microarrays. A protein microarray, also known as protein chip, is a high-throughput method which is used for determining the function, tracking of the interactions, and activities of the proteins on a large scale. In 1983, both the concept and the methodology of protein microarrays was introduced and demonstrated in antibody microarrays. The main advantage offered by protein microarray is the parallel tracking of a large number of proteins. The chip comprises of a support surface to which different types of proteins are bound. The support material may be a glass slide, a nitrocellulose membrane, a bead, or a microtiter plate. The probe molecules used for detecting the proteins in the array are labeled with a fluorescent dye. If there is a reaction between the probe and the immobilized protein, there is emission of a fluorescent signal which is detected by a laser scanner. It was relatively easy to develop protein microarrays because it is based on the technology that was used for the development of DNA microarrays. Protein microarrays offer the following advantages. They are rapid, automated, economical, highly sensitive, and there is consumption of small quantities of samples and reagents which makes it again highly economical. Reasons for development of protein microarrays. Due to the limitations posed by DNA microarrays, the protein microarrays were developed for the purpose of determining gene expression levels in proteomics. Often, the quantity of mRNA in the cell is not the true representation of the expression levels of the proteins. It is usually the protein and not the mRNA which plays a functional role in cell response. Thus, a novel approach was required. Post-translational modifications that are responsible for protein function are not visible on DNA microarrays. Protein microarrays have almost replaced traditional techniques such as 2D gel electrophoresis or chromatography because they are time-consuming, labor-intensive, and not well suited for analyzing proteins of low abundance. Types of protein microarrays. There are three major types of protein microarrays, namely analytical protein microarrays, functional protein microarrays, and Reverse phase protein microarrays. Analytical protein microarrays. It is also known as capture arrays. In analytical microarrays, a library of antibodies, aptamers, or Febodies is arranged on a solid support surface, which are used to capture molecules as each of them binds specifically to a particular protein. The array is probed using a mixture of protein solution like a cell lysate, and the resulting binding reactions can be detected using various detection systems. Analytical microarray gives valuable information regarding the expression levels of particular proteins in the biological sample and provides measurements of binding affinities and specificities. It is particularly useful in comparing the protein expression in different biological solutions. For example, a cell response to a particular factor is easily identified on comparison of the cell lysates of treated with specific substances. Another important application of this technique is in the identification and profiling of diseased tissues. Functional protein microarrays. They are also known as target protein arrays and are constructed by immobilizing purified proteins to the solid support surface. Functional microarrays differ from the analytical microarrays as they are composed of full-length functional proteins or protein domains that are used for studying an entire proteome's biochemical activities in a single experiment. It is useful for the identification of protein-protein interactions, protein-DNA interactions, protein-RNA interactions, protein phospholipid interactions, protein small molecule interactions, for assaying enzymatic activity, and finally for detecting antibodies and demonstrating their specificity. Reverse phase protein microarray. It involves complex samples like tissue lysates, for which the cells are isolated from the tissue of interest and then subjected to lysis. The lysate is arrayed on a solid support, is probed using antibodies against the target protein. The antibodies are detected using either chemiluminescence, fluorescent, or colorimetric assays. To perform protein quantification of the sample lysates, the reference peptides are printed on the slides. To perform protein quantification of the sample lysates, the reference peptides are printed on the slides. RPase is useful for the detection of altered proteins or other agents produced due to disease. Post-translational modifications are particularly altered as a result of disease and can be easily detected using RPase. Detection methods of protein microarrays. Detection methods for protein microarray must have two important properties. First, they should give a high signal, and second, a low background noise. Fluorescence labeling is the most commonly and widely used method of detection. It is highly safe, sensitive, as well as compatible with microarray laser scanners. The other labels that can also be used are affinity, photochemical, or radioisotope tags, which are attached to the probe itself. But these labels often interfere with the probe-target protein reaction. Detection methods of protein microarrays. Thus, several label-free detection methods have been developed. Some examples are surface plasmon resonance, carbon nanotubes, carbon nanowire sensors, micro electromechanical system, cantilevers. The above-mentioned label-free detection methods are comparatively new and unsuitable for high-throughput protein interaction detection. But they, however, hold much promise for the future. Applications of protein microarrays. The five major areas where protein arrays are being applied are: first, in the field of diagnostics. It involves first the detection of antigens as well as antibodies in the blood sample of patients. Secondly, the profiling of sera in order to discover new disease biomarker. Thirdly, in monitoring of the environment and the food samples. Protein microarrays are also important in proteomics. It refers to protein expression profiling where the proteins are expressed in the cell lysate. Protein functional analysis. It involves the identification of protein-protein or protein phospholipid interactions, small molecule targets, enzymatic substrates, particularly for kinases and receptor ligands. Antibody characterization. It involves the characterization of cross-reactivity, specificity, and mapping epitopes. Treatment development. It involves the development of antigen-specific therapies for the treatment of autoimmunity, cancer, and allergies. Secondly, identification of small molecules which can be developed as new drugs. Challenges in the development of protein microarrays. Several companies have made substantial investments in developing protein chips, but till now they have not flooded the market. The reason being that manufacturers find it difficult to handle proteins. Also, several steps are required for the manufacture of protein chip in comparison to a DNA chip. The challenges are: first, to find both a surface and a method of attachment which will allow proteins to maintain their secondary or tertiary structure. This will in turn help in maintaining their biological activity. To produce an array that has a long shelf life to prevent the proteins on the chip from denaturing. Identification and isolation of antibodies for every protein encoded by the human genome. Sensitively quantifying the levels of bound protein while avoiding the background noise. To be able to extract the detected protein from the chip so it could be further analyzed. To reduce the non-specific binding of the capture agents to the proteins on the chip. To develop high-capacity chip sufficient enough to allow the complete representation of the proteome. And finally, usually the more abundant proteins mask the detection of less abundant proteins like the signaling molecules and receptors. Peptide microarrays. A peptide microarray is also known as a peptide chip or a peptide epitope microarray. It comprises of a collection of peptides that are displayed on a solid surface, which is normally a glass or plastic chip. Peptide chips are used in the field of biology, medicine, and pharmacology for studying the binding properties, functionality, and kinetics of protein-protein interactions. A peptide microarray consists of a planar slide which either have peptides spotted onto it or they are assembled directly on a solid surface by in situ synthesis. The peptides spotted onto the slide are a product of single batch and can undergo quality control such as mass spectrometer analysis and concentration normalization before they are spotted. Whereas the peptides synthesized directly on the surface suffer from two disadvantages. The first being batch-to-batch variation and second is limited quality control options. But an important advantage of in situ peptide synthesis is the simultaneous synthesis of thousands of peptides. This helps in creating larger peptide libraries along with lower synthesis costs. Working of peptide microarray. The working of peptide microarrays is quite similar to performing an ELISA protocol. Several copies of the peptides are directly linked to the surface of a glass chip, whose size and shape resembles a microscope slide. The peptide chip is then directly incubated with the biological sample of choice such as a purified enzyme or antibodies, cell lysates of patient or animal sera. After several rounds of washing, a secondary antibody is added, which is tagged by a fluorescent label that can be easily detected by a fluorescence scanner. The other methods of detection are chemiluminescence, colorimetric, or autoradiography. Advantages of peptide microarrays over protein microarrays. One can study epitope spreading as the detection of binding events is at the epitope level. It allows for flexible design, allowing the incorporation of post-translational modifications, sequence diversity, and non-natural amino acids. The assays performed rely on detection of linear epitope interactions. It has an extended shelf stability and is cost efficient. There is higher batch-to-batch reproducibility. Applications of peptide microarrays. First, the peptide microarrays are useful in studying different kinds of protein-protein interactions, particularly the ones that involve modular protein substructures called peptide recognition modules or protein interaction domains. Peptide microarray is majorly used in the field of immunology for mapping of immunodominant regions in antigens or in complete proteomes, the profiling of changes in humoral immune response of individual patients during the course of disease progression, seromarker discovery, the monitoring of clinical trials, finding the neutralizing antibodies. Peptide microarrays is used in enzyme profiling for the purpose of identification of substrates for orphan enzymes to optimize the known enzyme substrates. It helps in revelation of signal transduction pathways. It helps in the detection of contaminating enzyme activities. It is used for determining consensus sequence and key residues. It helps in identifying sites for protein-protein interaction within a complex. Antibody microarray. An antibody microarray, also called as antibody array, is a type of protein microarray. It contains a collection of captured antibodies that are spotted and fixed on a solid surface like glass, plastic, or silicon chip used for detecting antigens. Say Wen Chang in 1983 gave both the concept and the methodology of antibody microarray while he was working in Centocor International in Malvern, Pennsylvania. The term antibody matrix was coined by Chang. He demonstrated that 100 and 400 antibody spots could be spotted on a 1 by 1 centimeter surface. He estimated that if an antibody is coated at a concentration of 10 microgram per ml, then 1 mg of antibody is enough to make 2 lakh dots of 0.25 millimeter diameter. Chang focused on the use of antibody microarrays for both the detection and quantification of cells that contain surface antigens like CD antigens and HLA allotype antigens, particulate antigens like viruses and bacteria. Thirdly, soluble antigens. There are several ways to create and detect antibody microarrays, and performing an antibody microarray is quite similar to performing an ELISA, the simplest being fluorescence-linked immunosorbent assay, which involves the use of an antibody chip to which cell lysate is added, which has the antigen in question, which goes and binds to the specific antibody on the chip. After this, washing is carried out to remove the unbound antigen or the other components of the cell lysate. After this, an detection antibody is added, which binds to the target antigen. Now, in order to detect this, a secondary antibody, which is fluorescently labeled, is added. This binds to a specific epitope on the detection antibody, and upon excitation with a laser light gives the appropriate fluorescence, which is subsequently detected. Applications of antibody microarray. It is useful in research for the detection of protein expression, particularly from cell lysates. It also has diagnostic application as special biomarkers from serum or protein. It is used for profiling experiments on samples obtained from tissue, plasma, serum, etc. Antibody microarray based profiling studies are used for studying and detecting cancer. For example, in 2010, an antibody microarray containing 810 different cancer related antibodies was developed and used for studying cancer. Tissue microarrays. Tissue microarrays are also called as TMAs, and they consist of paraffin blocks in which up to 1,000 separate tissue cores are assembled in an array fashion, which enable multiple histological analyses to be performed simultaneously. The burdensome nature of procedures, limited accessibility of diagnostic reagents, as well as the limited patient sample size are some of the limitations that affect molecular clinical analysis of tissues. Thus, the technique of tissue microarray was developed to overcome these problems. Applications of tissue microarray. H. Battifora in 1986 was the first to introduce multi-tissue blocks, which was known as the multi-tumor sausage tissue block. It was later modified in 1990 and was known as the checkerboard tissue block. It was in 1998 that the current technique was developed by J. Kononen and collaborators. They made the use of a novel sampling approach which led to the production of tissues of regular size and shape which could be more densely and precisely arrayed. Tissue microarrays are mainly used for the analysis of cancer samples. Procedure for preparing tissue microarray. First, a hollow needle removes tissue cores from the regions of interest in a paraffin-embedded tissues obtained from clinical biopsies or tumor samples. Secondly, the tissue cores are inserted in a recipient paraffin block but in a precisely spaced array pattern. These blocks are used for sectioning using a microtome, and each microarray block can be cut into 100 to 500 sections. Each of the tissue section can be subjected to independent tests and then analyzed by a standard method of histological analysis. Immunohistochemistry and fluorescent in situ hybridization are the two tests that are commonly employed in tissue microarray. Cellular microarray. A cellular microarray is used for examining living cells on the surface of a solid support called a chip. The chip is spotted with antibodies, proteins, or lipids that interact with the living cells. This in turn leads to their capture on specific spots on the chip. Combinations of antibodies, proteins, and lipids can be spotted in a given area that leads not only to the specific cell capture, but if there's a specific interaction, it will lead to first, stimulation of a cellular response, second, change in phenotype, third, detection of a response from the cell like secretion of a specific factor. There are two major types of cellular microarrays. First is reverse transfection cell microarrays. Second is PMHC cellular microarrays. Reverse transfection cell microarrays. It was developed by David M. Sabatini's laboratory at the Whitehead Institute in 2001. The DNA is printed on a glass slide to facilitate the transfection process, which is the deliberate introduction of nucleic acids into cells. Next, the cells are added to the array and incubated. Since the order of addition of DNA and adherent cells is reversed in comparison to that of conventional transfection method, this microarray technique is called reverse transfection microarray. Purified DNA plasmid is mixed with the gelatin solution, which is used for printing onto a slide. Besides gelatin, one can use etylocollagen and fibronectin as transfection vectors to introduce foreign DNA into cell nucleus. This technique allows for high throughput screening as hundreds of genes may be expressed in cell on a single microarray. Thus, it is useful for studying gene expression and regulation. PMHC cellular microarrays. It was developed by Daniel Chen, Yov Swan, Dan Kraft, Patrick Brown, and Mark Davis at the Stanford University Medical Center. These cellular microarrays are spotted with peptide MHC class 1 or peptide MHC class 2 complexes. These biochips are used for interrogating the immune cells, mainly the antigen-specific T cells, from clinical samples to know what they are capable of recognizing. They can be co-spotted with other molecules like antibodies, which can capture cytokines, thus allowing the functional analysis of the captured T cells. Chemical compound microarray. A chemical compound microarray consists of a solid surface like a glass and plastic spotted with a collection of organic chemical compounds. Its format is similar to that of DNA, protein, or antibody microarray. It has been developed in chemical genetics research for the following purposes. First, to search for proteins that bind with specific chemical compounds. Second, for drug discovery research. Types of chemical compound microarray. There are three different types of chemical compound microarrays on the basis of their fabrication method. The first is small molecule microarray. The organic compounds are covalently immobilized on the solid surface using diverse linking techniques. It was invented and advanced by Dr. Stuart Schreiber and his colleagues. The second type of microarray compound screening. It was developed by the scientists in Abbott laboratories and involves spotting and drying organic compounds on the solid surface without the need of immobilization. Discovery dot technology. This microarray technique was developed by Dr. Scott Diamond. It was later on commercialized as Discovery dot technology by the Reaction Biology Corporation. Here, the organic compounds are spotted from a homogeneous solution without the need of immobilization and drying. The phenotype microarray. The phenotype microarray is used for the high throughput phenotyping of cells. It enables simultaneous monitoring of the phenotypic reaction of cells to first, environmental changes, second, exogenous compounds. The recording of phenotypic reactions is either in the form of endpoint measurements or as respiration kinetics, which is similar to growth curves. For performing phenotype microarray, a sole carbon source is transported into a cell, followed by its metabolism to produce NADH. This generates a redox potential and also flow of electrons that reduces a tetrazolium dye like tetrazolium violet, producing the purple color. The greater and faster the metabolic flow, the quicker is the formation of the purple color. The formation of purple color is considered to be a positive reaction as the sole carbon source has been used as a source of energy. For performing the phenotype microarray, the following are required: a microplate reader, incubation facility, incubation chamber that provides the suitable incubation conditions and automatically records any change in the intensity of color. Fourth, software to analyze and interpret the data. The information regarding the capability of an organism to use certain energy sources can be equivalently applied to the use of other micronutrients like nitrogen, sulfur, phosphorus, etc. Secondly, it can further be used to assess the impact of oxotrophic supplements, antibodies, heavy metals, and certain inhibitory compounds on the cell's respiration behavior. Applications of phenotype microarray. Gaining information regarding the biology of bacteria, fungi, yeast, and animal cell lines like the human cancer cells. Collectively and quantitatively measures thousands of cellular phenotypes. Testing the gene function and for improving genome annotation. It is performed with live cells, which providing comprehensive information regarding the performance of entire cells. Used in the fields of mammalian cell physiology, including clinical research, systems biology, microbial cell physiology, and taxonomy. Advantage of using phenotype microarray over standard growth curve is that it makes it possible to measure cellular respiration in environmental conditions where cellular replication might not be possible, and the respiration reactions are generally detected much earlier than cellular growth. Carbohydrate microarray. The concept of carbohydrate array was given by Ten Fizzi and co-workers in 1985. The oligosaccharides are immobilized on a solid surface to study their interactions and or recognition by antibodies in certain carbohydrate recognizing proteins. The carbohydrate microarray involved the release of oligosaccharides from glycoproteins followed by their conjugation to a lipid molecule which converts them to neoglycolipids (NGLs). The neoglycolipid-based oligosaccharide microarray was developed in 2002, containing sequence-defined oligosaccharides. It has been useful for demonstrating that oligosaccharides are involved in diverse molecular interactions and that either directly or indirectly they are involved in majority of diseases. By coupling the NGL technology with mass spectrometry, carbohydrate microarray has become a powerful tool for allocating the epitopes of monoclonal antibodies directed at carbohydrate differentiation antigens. Second, receptors for bacteria. Third, recognition of ligands of numerous carbohydrate recognizing proteins belonging to the innate immune system. So students, let us now summarize what we have learned in this module. Firstly, a protein microarray is a high throughput method used for determining functions, tracking the interaction and activities of proteins in parallel on a larger scale. The chip comprises of a support such as a glass slide, a nitrocellulose membrane, bead, or a microtiter plate to which different types of proteins are bound. The probe molecules used for detecting the proteins in the array are labeled with a fluorescent dye which emits fluorescent signal upon hybridization. Protein microarrays were developed due to certain shortcomings of DNA microarrays. Although protein microarrays are rapid, automated, economical and highly sensitive, but still they are not commercially successful in comparison to DNA microarrays. Peptide microarray comprises of a collection of peptides displayed on the surface of a solid surface such as a glass or plastic chip. Peptide chips are useful in the study of binding properties, functionality and kinetics of protein-protein interactions. An antibody microarray is a type of protein microarray containing a collection of capture antibodies spotted and fixed on a solid surface like glass, plastic or silicon chip. It was developed by Se-Wen Chang in 1983 for detecting antigens. Presently, the antibody microarrays have diagnostic applications, are used in research for the detection of protein expression, particularly from cell lysates, and in profiling studies for studying and detecting cancer. Tissue microarrays, also known as TMAs, are composed of paraffin block containing up to 1,000 separate tissue cores assembled in an array fashion. This allows multiple histological analysis to be performed simultaneously. A cellular microarray is a unique technique for examining live cells on the surface of a solid support called chip, which is spotted with antibodies, proteins, or lipids. These biomolecules interact with the living cells, leading to their capture on specific spots on the chip. A combination of these molecules can also be spotted, which leads not only to the specific cell capture but If there is a specific interaction, it would lead to either stimulation of a cellular response or a change in phenotype or a secretion of a specific factor. A chemical compound microarray is composed of a solid support like glass or plastic, which is spotted with different types of organic chemical compounds. It has been developed in chemical genetics research to look for proteins that bind to specific chemical compounds and for conducting research in drug discovery. The Phenotype Microarray is a high throughput method that has enabled simultaneous monitoring of the phenotypic reaction of cells to environmental changes or exogenous compounds. The recording of phenotypic reactions is either in the form of endpoint measurements or as respiration kinetics, which is similar to the growth curves. Lastly, the carbohydrate microarrays involve immobilizing the oligosaccharides on a solid surface to study their interaction or recognition by antibodies and certain carbohydrate recognizing proteins.